Hey everyone, this mini lecture's on biochemistry. We've been looking at things like organic molecules, the alkanes, the alkenes, the alkynes, etc. In one way, I guess, one branch of organic chemistry is biochemistry, and clearly that's dealing with the chemistry of life itself. In this particular mini lecture, we're only going to look at one type of biomolecule, the proteins, and I guess the constituents of proteins, which are amino acids. The reason, of course, is that we just simply don't have time to cover much more than that. But it's a good way of illustrating the relevance of chemistry and organic chemistry to the study of the chemistry of life itself. So, as I said, one of the groups of organic molecules are biomolecules are the proteins. Proteins are responsible for carrying out just about every activity that occurs in living things. They're really the workhorses of cells. I know I've talked a little bit previously about DNA, but DNA is really carrying the code for life, but it's the proteins that are actually carrying out the activities of life itself. They make up things like muscle tissue, they're responsible for motility, that is movement, and they're also enzymes, that is they're biological catalysts. And I talked a little bit about the action of catalysts in a previous lecture, but basically enzymes are these proteins that are catalysts, and they speed up reactions without actually being consumed in the reaction itself. One important thing about proteins is that they fold into particular shapes. They're made up of subunits we call amino acids, and I'll talk about them a little bit in a moment. But the important point here is that the particular shape that a protein folds up into then actually determines the function of that protein. If the shape isn't right, the protein just simply won't function properly. And I'll talk a little bit about that again in a moment. That folding, as you can imagine, is very important to protein function. I've just pointed that out. But what it really depends on then is the charge on the side chains. What side chains am I talking about? Well, these are the chains of amino acids. They've got side chains associated with them. And some of those side chains pick up charges, and those charges then determine how the whole protein folds together. And the important point about that then is that pH actually has a profound effect on the charge of the side chains. So as the pH goes down, you've got more and more protons in solution. Those protons can combine with bases, like for example is one that's negatively charged called carboxylic acid, and it turns it from a negatively charged carboxylic acid to a neutrally charged side chain. And again, this has a real effect on protein function. So the bottom line with all of this is that proteins are extremely sensitive to changes in pH. So it's not surprising then that it doesn't actually take much of a change in pH to disrupt protein function. Okay, I was talking about these things called amino acids. What are they? Well, if I just get my pen here, just to illustrate a little more detail. The common function between these amino acids is that they have this central carbon here, attached to which are these two groups. This is called a carboxylic acid group, and this is called an amino group. Hence the name for these things, amino acids, and you can see that they all have these amino acid groups attached to them. Now, what differentiates these amino acids are these side chains I was talking about. So here's a side chain here, here's a side chain here. Now you can see here that these side chains in this particular group of amino acids are just hydrocarbons, and therefore they're hydrophobic. That is, their bonding is mainly due to van der Waals forces. These amino acids here have hydrophilic side groups, that is, they can bond with water. And these amino acid groups here, they're kind of a mixture, if you like, but some of them can bond with water and some can't. Now, my point again here is that what differentiates these amino acids are these side chains. If you count these up, you'll find that there's 20 of them. So all proteins, and there's literally hundreds of thousands of different types of proteins, are just made up of combinations of these 20 amino acids. And they're often relatively large molecules, these proteins, usually made up of hundreds of amino acids joined together. So you can see there's many, many different combinations. Okay, so one of the things with proteins is because they form such complex shapes, often you don't draw them with all the individual atoms involved because it would just be simply too complex to work out what's going on. But what you can do is use these kind of what are called cartoon diagrams here, or ribbon diagrams, to show how the amino acids are joined together and the sorts of shapes that they form. So in this case here, this particular protein has got this thing that looks like a helix, and in fact it is, and it's called an alpha helix. And you can see now that they're actually made up of this protein's made up of quite a few alpha helices. There's one there where it's orientated so that you're looking down the barrel of the helix. There's one here, there's one here. Let me get my pen. So there's the barrel there. We've got one here, we've got one here. This part here of the protein is called random coil. There's actually nothing random about it. The amino acids are forming a particular shape here as well, but it doesn't have this regular repeat structure like it does with these alpha helices. This stuff here, we've got these two arrows running anti-parallel to one another. That's actually beta pleated sheet. And again, I don't have time really to go into what these look like in great detail. I just want to again point out that proteins form different shapes, and that's what gives them function. Now, the next thing I just want to point out to you is if you understand the importance then of shape with proteins, what do proteins actually do to carry out function? Well, one of the most important groups of proteins are the enzymes, and they act on particular small molecules to change them in some fashion. So for example, in this case here we've got kind of a diagram representing an enzyme in this case. I'll get my pen here, so here's my enzyme here, and it has a shape where it can actually attach to the thing it's going to act upon, called the substrate, and then convert it in some fashion. Now the important thing to understand here is that the two shapes are complementary to one another. If the enzyme doesn't have this shape here, then it won't fit in with its substrate, and therefore it can't act upon its substrate. So again, you can see that the structure of an enzyme really does determine its activity here. People often refer to this as the hand in glove model. So if you can imagine this here, the substrate is the hand, it can fit into this thing here, which is the glove. And fit very nicely, it does too. Now, because I didn't really mention the term active site, that particular area here that fits very nicely with the substrate is actually called the active site. So my major point here is that substrates fit active sites, and they fit them like hands in gloves, almost literally. And you know this, you can't put your left hand into a right-handed glove, well, not very easily anyway, unless you force it, and if it's made of quite stiff leather or canvas or whatever, it's impossible to do. So whilst the two gloves look identical, when you're looking at left and right-handed gloves, they in fact aren't. They're actually mirror images of one another. So this is an important point. This means that when you're looking at a drug acting, and a drug is just normally a small molecule that has some sort of an effect on enzyme or protein function, the shape of the drug is important, but also the mirror image type of the drug. let me illustrate what I mean again by example. So here's an example here, and I've just started off trivially here with a baseball glove, and here's a mirror image of the same baseball glove here. And you can see on first inspection you might think that they're identical, but they're not. They're mirror images, and you certainly can't put your right hand in a left-handed baseball glove and vice versa. If we look at this drug here, and it's actually the bottom one that's the drug, it's got a particular shape, and it will fit into a particular active site in an enzyme, but it does have a counterpart which is a mirror image. Yet oddly enough, it's only this one here that's active. This one is completely inactive. So you can see that this understanding of chemicals and the idea that you can get different mirror images of chemicals is extremely important in things like the pharmaceutical industry. I just want to introduce one other term to you here. When you're looking at molecules and their mirror images, they're referred to as stereoisomers of one another. Okay, that's it for this particular lecture. That's it really for the series. I hope you enjoyed it. Bye.